In this screencast video lecture, we are going to see in detail about the nitrogen fixation. It refers to conversion of molecular nitrogen that have been present there in the atmosphere into various nitrogenous compounds such as nitrite, nitrate and even ammoniacal form. And this nitrogen fixation could be taking place through three different types. First one is atmospheric nitrogen fixation, second one is an industrial nitrogen fixation, and third one is referred as a biological nitrogen fixation. First two things are associated there with the abiotic way of fixing the nitrogen, whereas the third one, that is biological nitrogen fixation, shortly referred as a BNF, mainly involves conversion of nitrogen in the atmosphere into ammonia molecules, which is mediated by the nitrogenous enzyme that was present there in the bacteria. And this property is primarily associated there with the prokaryotic organisms alone. That is, none of the eukaryotes can able to fix the atmospheric nitrogen into ammonia. Now we look at the details of the different kinds of nitrogen fixation. The first one is atmospheric nitrogen fixation. It involves conversion of molecular nitrogen into nitrogenous compounds through lightning or meteorite trials or cosmic radiations. This lightning, meteorite trials as well as cosmic radiation provide the necessary energy that is required for the physicochemical conversion of nitrogen into various nitrogenous molecules. It takes place in a stepwise manner. In the first step, nitrogen on combining with the oxygen can able to form into nitric oxide. This nitric oxide again combined with another molecule of oxygen can able to form nitrite. Nitrite that have been present there in the atmosphere can able to get easily washed with the rainwater and it forms into nitrous acid or nitric acid. Sometimes when the nitrous or nitric acid are coming into the ground through the rain process or through rainwater, they can react with certain alkali radicals to produce some derived salt such as a calcium or potassium nitrate salt that may be present there in the soil system. Next, we look at the points related to the industrial nitrogen fixation. It's a man-made process in which the molecular nitrogen is converted into nitrogenous usable form. And this particular process has been invented by Haber-Bosch and it is referred as a Haber-Bosch process of nitrogen fixation. Here, the operation of this process requires high temperature, pressure and a lot of catalysts such as ruthenium or iron is required to convert the nitrogen into ammonia. So this process takes place in the presence of nitrogen and hydrogen that are combined to form into ammonia in the presence of a high temperature and pressure. Here the temperature could be around 400 degrees centigrade, whereas the pressure could be elevated to 200 atmosphere pressure. Under this condition, nitrogen has been converted into ammonia. This is a way in which the ammonia fertilizers that are commonly used there in the agriculture are all produced there in the industry. The next one is the biological nitrogen fixation, which is shortly referred as a BNF process. It refers to the conversion of molecular atmospheric nitrogen into a usable form of nitrogenous compound, mainly the ammonia. This takes place by the intervention of the bacteria and archaea and it is referred as a biological nitrogen fixation or BNF. The organisms that have been involved in this process are technically referred as a diazotrophs. Diazo meaning nitrogen, nitrogen utilizing organism. That is organism that converts the nitrogen into ammonia. This BNF process accounts to about 100 to 140 teragrams of nitrogen that have been fixed into ammonia per year. This process again involves nitrogen and hydrogen that are combined into ammonia molecules, whereas here instead of a high temperature and pressure that have been employed there in the industrial process, only energy in the form of ATP and this whole process is mediated by the catalyst that is the nitrogenous enzyme system. Here the temperature is very ambient, that is around 20 to 30 degrees centigrade could be favoring the nitrogen fixation, and the atmospheric pressure is also of a normal atmospheric, that is 1 atmospheric pressure at which this whole process could be easily completed. So compared to the industrial nitrogen fixation, This process is totally a biological and sustainable process which is mainly mediated by the bacteria. Now we try to understand the different types of bacteria that may be involved there in this particular process. There is a wide variety of phylogenetically different groups of bacteria and physiologically different forms of bacteria have been involved there in the nitrogen fixation process. Some bacteria can able to use sunlight as an energy. They are all phototrophs, and they can able to use further the reduced inorganic elements as electron donor, that is a lithotrophic in nature, whereas other groups of bacteria can able to use different kinds of organic compounds that are referred as a heterotrophic group of bacteria. Again, based on the oxygen requirement, certain nitrogen fixers are obligate aerobes, some are facultative aerobes, whereas some are obligate anaerobes. We look at the examples of the different group of organisms there in the right hand side. Again, based on their habitat, that is the place in which they are living, they can be classified as a free living bacteria, associative bacteria, and a symbiotic bacteria, and free living anaerobic group of bacteria. Here, free living aerobic bacteria is the Acetobacter, Behringia and Klebsiella, and the cyanobacteria that have been present in the various environments are coming under the aerobic group of nitrogen fixing organism. Whereas the free living associative symbiotic bacteria is a very classical example is Azoospermum. Whereas obligate symbiotic bacteria example are Rhizobium, which commonly lives there on the legume plants, and Frankia, which is commonly associated there with the alder plants. Free living anaerobic bacteria, very classical example is a Clostridium. Now we try to look at some points related to the symbiotic nitrogen fixation. As I already told, the classical example is the Rhizobium legume nodulation. Here they specifically use a term called as a cross inoculation group. It refers to classification scheme that is used to designate the group of forage legumes that have been inoculated and successfully nodulated by a particular species or a biovar of Rhizobia. Very simple example is the alfalfa plants, which are commonly nodulated only with the help of Rhizobium meliloti. That is, one particular Rhizobium specifically found associated there with one particular type of a legume and can able to successfully nodulate it. So this particular Rhizobium is referred as a cross inoculation group for that particular legume plant. So based on that, if you look at, these are the various cross inoculations group that have been identified so far. Say for example, the first one is referred as a P. Rhizobia, which mainly belong to the Rhizobium leguminosarum biovar vitae group, which can able to nodulate the peas, witches plants, lentils, and even the faba bean. The next one is a bean Rhizobia cross inoculation group, which is referred to Rhizobium leguminosarum biovar facioli, which can able to mainly nodulate the bean plants. That is, they cannot able to nodulate the peas. Thus, a stringency there in the nodulation of a particular host plant can be easily seen. The next one is a clover Rhizobia, which is referred as a Rhizobium leguminosarum biovar trifoli, which can able to nodulate only the clover plants. The next one is alfalfa Rhizobia, which can able to nodulate only the alfalfa. That is a Medicago species. Then like that, the other groups are chickpea Rhizobia, which will be nodulating only the chickpea. Then soybean Rhizobia. Then Leucana Rhizobia, which belongs to a Rhizobium species, which can able to nodulate the various kind of a leucane plants. Finally, the cowpea Rhizobia group, which belongs to Bradyrhizobium genus, that can able to nodulate the different cowpea group of plants, which includes the pigeon pea, peanut, then cowpea, black gram, then lima bean. So these are all the various plants that could be successfully nodulated by this particular cross inoculation group.